Hi, welcome back to video 2. We are going to discuss mitosis and the cell cycle, along with cancer. All right, so after talking about DNA replication, and we, have it down that the cell needs to replicate its DNA before it's going to divide so that those new daughter cells have a complete set of genetic instructions. Now we're going to talk about when do cells actually divide. So, and why do they divide? Well, number one, over here, cells are going to divide for growth, right? You did not come into the world the size that you are now. It took your cells dividing, growing and dividing over multiple years for you to reach that size. So clearly, organisms need to go through cell division for growth. They also need to go through cell division for cell repair. You might break a bone, you might get an injury to your skin, maybe you have surgery. All of these things are going to require a repair of the damage. So we need to divide and make more cells to repair that damage. Cells are also going to go through mitosis for replacement. Your cells do not live forever. They age, they acquire mutations, and part of the natural process to maintain healthy cells is to have something called like a cell turnover. In fact, about every 2.5 weeks, the top layer of your skin has completely replaced itself. Red blood cells can regenerate about every 2 months or so, 6 to 8 weeks. We can have, a new set of red blood cells. So cells are constantly going through a repair and a replacement process that requires mitosis. Cells also use cell division to maintain an efficient cell size. If you remember when we talked about the cell membrane and the fact that cells need to exchange materials across the cell membrane in an efficient manner so that they can have those materials to do their cell processes quickly. So cells, if you remember, always want to have a large surface area, but a very small volume. So you're not going to have a cell that grows infinitely big. Once a cell reaches a particular size, it's going to divide to maintain that efficient cell size. Now, one thing that I didn't put on this slide is that most of this stuff right here, I should say all of it, applies to multicellular organisms. Unicellular organisms like yeast, bacteria, protists, they're going to use cell division for continuation of their species, right? If we want to go from one bacteria cell to two bacteria cells to an entire colony of bacterial cells, then cell division is going to actually duplicate an entire organism. Not just what we call the cell, because that one cell is the entire organism. So the cell cycle. All right, so the cell cycle is a continuous process of growth and division that all cells go through. It is a highly regulated process. There are checkpoints along the way. A cell is constantly asking itself, do I have the right materials? Am I big enough? Is all of my DNA copied? Are there any mutations? Do I have enough organelles? Do I have enough proteins? Do I need to make more proteins? It is a highly regulated process. It's not just like free willy-nilly cells are just dividing whenever they want to. We'll get to what happens when that's the case. Now, the cell cycle is made up of three main components, and those three main components are listed here on the right-hand side. We have interphase. Interphase is the preparation phase. It's subdivided into G1, S, and G2. G1 is all about cell growth. The S phase is when replication of DNA takes place. G2 is more growth. It's also going to replicate some proteins and make some more organelles because remember, this one cell is going to divide into two, so it has to make sure it has enough proteins and organelles to go around. Step 2 of the cell cycle is mitosis. Now, another name for mitosis is karyokinesis, which simply means division of the nucleus or division of the genetic contents. And then last but not least, we have cytokinesis, which is division of the cytoplasm. And cytokinesis is where we actually get two distinct daughter cells. Here's the diagram of the cell cycle. And you can see we can start right over here. I'm going to start here with this teal colored arrow. So the teal colored arrow, basically all of this stuff in like this light gray here, these um, bottom 3/4 of this wheel, this is all interphase. You can see that here it's represented in the dark blue. So here's G1, here's S, and here is G2. And there are checkpoints along the way. In fact, there's a really, really important checkpoint right here after DNA replication called the S checkpoint. And what the cell does there is make sure that all of the DNA has been copied correctly. And if it's not, it's going to go through a cycle of what's known as programmed cell death or apoptosis. And you want that because you don't want a cell that has acquired a lot of mutations to go on and pass those mutations on to future generations of cell lines. That's how you acquire, you can, one of the ways that you can acquire mutations that might lead to cancer. D2, last bit of growth, last bit of preparation for mitosis, and then we actually go through the M phase. And you guys hopefully remember this, maybe from life science, but in mitosis, we have prophase, metaphase, anaphase, telophase. Now, the one thing that's left out here, which would be right at the end, would be cytokinesis. So let's take a little bit closer look at some of the actual rates of the cell cycle and the steps, things that are going on, okay? So the rate of the cell cycle, well, first of all, it depends on what the cell is. Cells like skin cells, they can divide rather quickly and you can replace your entire top layer of cells in about 2.5 weeks, right? Other cells don't divide as quickly. Red blood cells, for instance, don't divide as quickly. Bone cells, liver cells, these things all grow, they replace themselves and they repair themselves, but all at different rates. Um, some cells enter what's called a G0 phase, meaning after they go through an initial round of growth, they sort of enter what was used to be called as a resting stage, but it's not really resting because the cell is still doing metabolic processes, but it's just more of like a non-dividing stage. Your muscle cells are like this. In fact, you're born pretty much all the muscle cells you will ever have. Well, how do your muscles get bigger then, you might ask? When you're working them out, when you're lifting weights, doing athletic activities, your muscles get torn up. You kind of put little micro tears in them. And your body's way of repairing that is to add protein to those muscles to help repair those tears. So muscle building, strength building is actually a process of breaking down the muscle when you're doing the lifting activities and then putting in protein to help repair that. The more that wear and tear you put on the muscle, the more you're telling the body that it needs to build that muscle up. So the bigger that it gets. Nerve cells. In the brain and throughout the body typically do not go through division. They don't regenerate themselves, which is why conditions such as paralysis or even strokes, if you have brain cells that are damaged from a stroke, right? We never fully get those back. Now, there is current research that says you can pull some of those nerve cells back. And there are current projects going on looking at that, maybe getting nerve cells to regenerate themselves. But for the most part, they're not dividing. All right, the end result of mitosis. So mitosis is all about it's a type of asexual reproduction, right? Which means we don't need sperm or egg cells. We're talking about one cell making a copy of its DNA and then dividing that DNA into two different daughter cells. So the end result of mitosis is one parent cell that is diploid. So the term diploid means to have a complete set of chromosomes, two sets of chromosomes. In the terms of humans, a complete diploid set would be 46 chromosomes. We have a set of 23 from mom and a set of 23 from dad. So if a human cell that has 46 chromosomes goes through mitosis, the two daughter cells at the end of mitosis will each have 46 chromosomes. If we have a human or any other, it doesn't have to be a human cell. Let me change that. What if we had a cat cell, right? If we had a cat cell that had 38 chromosomes and that cat cell went through mitosis, the two daughter cells would each have 38 chromosomes. So the end goal of mitosis is to go from one parent cell that is diploid to two daughter cells. that are diploid. We're keeping the genetic um, disposition of the cells all the same, right? And think about that. Think about why we're going through mitosis in the first place. We're going through mitosis to repair cells, to replace cells. If you break a bone and you need to heal that bone material, you certainly want it to be repaired with the same cells that you lost in the first place. You don't want anything to be different. So we're making genetically identical cells at the end of mitosis. And there we just talked about um, the number of chromosomes in the human body. So let's look at the actual phases of mitosis here. So we have interphase, preparation for division, it's regulated by checkpoints, etc. Now, during interphase, and here's what a typical cell looks like in interphase, the DNA is real loosely coiled up in here. It's not arranged in chromosomes just yet. This loosely coiled DNA is referred to as chromatin. Now, once DNA replication takes place, and that DNA becomes organized into chromosomes, which is these packages of genetic information that can be neatly distributed between the cells during cell division. We end up with, I'm going to jump to here. We end up with two identical halves of a chromosome, two that are known as sister chromatids. So every chromosome, once it's, a chromosome is a condensed package of DNA wrapped around proteins that help keep it all in place and help it keep it compact inside the cell. When that chromosome goes through replication, so here's our teal color or our green color and our purple color. When each one of these chromosomes goes through replication, it creates an exact replica of itself called a sister chromatid. Now, together, the pair is called a homologous chromosome pair. Homologous chromosomes are chromosomes that are of the same size, the same shape, and they have the same genetic information. Typically, we're talking about chromosomes where one chromosome of the pair came from mom, one chromosome of the pair came from dad. But this is the chromosome structure that you guys are used to calling a chromosome. It's this X-like shape. But again, this is a replicated chromosome. So when you see those things in that X-like shape, this is a replicated chromosome. It's made of two sister chromatids. They're the exact same genetic information. And this is how we see chromosomes when we look at a cell in prophase. Prophase is the first step of mitosis. And here in prophase, we can see our chromosomes have condensed. So we see those actual X-like shapes. The centrioles, which are represented, they're not drawn in here real nice, but these little starbursts here at each corner of the cell, that's the location of a special organelle called a centriole. And the job of the centriole is to make spindle fibers. Spindles are proteins, and the job of spindles is going to be to attach to the chromosomes and help separate them during anaphase and telophase. Over here, after prophase, right, we have our chromosomes condensed, we have our spindle fibers form. Those spindles start to attach to the chromosome. So all of these dashed lines are the spindle fibers, and you can see they are attached to the chromosomes, or more specifically, they're attached to each sister chromatid. Now, metaphase, I like to think M for metaphase, M for middle. What does that mean? Well, that means that all of my chromosomes you can see are lined up in the middle of my cell. So spindles are attached to the chromosomes. Chromosomes are moved to the middle of the cell. There is a checkpoint here, a really important checkpoint. And the checkpoint is this: are all of the chromosomes attached to a spindle fiber? The job of the fibers is going to be to pull these chromosomes apart so that we get an equal amount of chromosomes on either side of the cell when it finally divides into two. If one of these chromosomes fails to attach to a spindle fiber, you're going to end up with a cell that has too many or too few chromosomes. Either way, it's going to be a cell that has a mistaken chromosome count and probably will go through that programmed cell death and not even survive to grow again and divide. So once all the chromosomes are lined up in the middle, here comes that role of the spindle fibers. So the job of the spindle fibers is to pull the chromatids apart, away from their partners. A for anaphase, A for apart. So you can see our chromatids are being separated to opposite sides of the cell. Lastly, telekinesis and cytokinesis. So telophase, I think I just said telekinesis, you can ignore that. Telophase is the last step of mitosis, and then cytokinesis is the last part of the cell cycle. So telophase, we have two cells, I like to think of it that way. We have two nuclei being formed around our chromosomes, and then eventually cytokinesis, the cytoplasm is going to divide. Now, in cytokinesis, takes on a couple of different looks depending on whether or not we're talking about an animal cell or a plant cell. In an animal cell, you can see this little groove, this little furrow. That is called a cleavage furrow. And you can go ahead and take a couple seconds to laugh. Everybody does. But this is called a cleavage furrow, and it's just a little groove, a little indentation in the cell membrane where cytokinesis is going to finally divide these two cells into two daughter cells. Now, in a plant cell, we can't do that. We can't like pinch in the cell membrane because there's a cell wall surrounding a plant cell. So in plant cells, we have to form a cell plate. I don't have a picture of it, but a cell plate is sort of like a juvenile cell wall. It's the beginning, the cell starts to lay down some cellulose and just starts to make a very premature cell plate that will eventually mature into a cell wall and create two final daughter cells. Just to wrap up, mitosis is asexual reproduction. So no sperm or egg cells. Daughter cells are basically clones of the parent. They are genetically identical. And I mentioned this before. If you're a unicellular organism, you're using mitosis to continue your species. Yeast, for example, right here, they're going to, they use a process called budding. So you can see right here is a mature yeast cell, the larger cell, and then that little guy coming off of it, that's his bud, which will eventually grow big enough to become its own individual yeast cell. Bacteria go through a process called binary fission to make more bacterial cells. Hydra, over here in the bottom left, they do budding as well. So here's the adult organism, and then this little branch coming off to him to the bottom right is another bud. All right, that wraps up the steps of mitosis, and that was a little longer than I thought it was going to be. So I will make a third video actually where we just talk about cancer a little bit. So stay tuned.